So now we're trying to connect via Zoom to the next speaker, who is Anjali Goswami from the Natural History Museum in London, with a title, From Development to Deep Time, Reconstructing the Evolution of Diversity with a Phenomic Approach. Are you on?
Yeah. I am. Can you hear me?
Yes. Thank you for being here. We're listening.
Okay, sorry for the mix-up. Perfect. Okay, thank you very much for the invitation, and I'm very happy to be able to join you remotely, although of course I wish I could be there in person. Hopefully, I'll still be able to share with you some of the research we've been doing in my group over the past few years, or actually over the past few decades, actually, looking at the evolution of morphological diversity across really the full breadth of land vertebrates or tetrapods, and trying to bring multiple perspectives into our understanding of how different morphologies evolve and how morphological diversity evolves. And I think we can all appreciate that the evolution of diversity is really a fundamental topic in biology. It's really important for understanding how we generate the diversity on the planet today, but also for understanding how that diversity will change in the future as we go through quite an extreme environmental change. And for those of us who think about diversity on these really large scales, it's quite obvious right away that diversity is not evenly distributed. So if we look across the Earth today, we can see that there are many more species in the tropics and in the poles, such as in this figure here, where we have the really red spots representing areas of high diversity. If we look through time, we can see periods in the past where we had relatively high standing diversity, such as in the Paleozoic. And then we can also see this is punctuated by really rapid decreases in diversity with the 5 main mass extinctions, and also relatively rapid increases in diversity, usually in response to these extinctions. And finally, when we look across the tree of life, we can see that there's a big variation in the diversity of different groups or clades of organisms. So we can see that some groups, say, birds, are hugely diverse in species numbers, while other groups like crocodiles have relatively few species. And so the question that I've always been very interested in is trying to understand what processes shape organismal diversity over these kinds of large scales. Why do we see these differences in how diverse organisms are, how diverse time periods are, how diverse different regions are? Now, all of those examples that I just told you about are talking about species diversity, how many species are in these different bins that we are looking at. But there are, in fact, many ways to measure diversity. And the way that I think about diversity is actually quite different. I don't think about species numbers. I think about diversity of form or diversity of anatomy, or what we would call morphology. More broadly, we can call this an organism's phenome. And a phenome represents their observable physical characteristics. And these are the characteristics that really mediate their interactions with other individuals, with other species, with the environment. And it's also really great because it allows us to reconstruct their ecology and their life history, even when all we have is their morphology, when we don't have great genomic data like what we were just hearing about. Now that's important because that really allows us to bring in the really, really rich data that's available in the fossil record. The vast majority of life on Earth is extinct. So if we look just at tetrapods, the land vertebrates, the vertebrates with 4 limbs, everything from amphibians to birds and mammals, there are twice as many extinct families as there are living families, and they represent a huge range of variation. So we want to be able to include those species and those forms in our understanding of how diversity evolves, as well as looking at living forms. And this, of course, is important because it's not just species numbers that are extinct, but actually the vast majority of variation in form is extinct. So if we think about, the ancestors of birds as dinosaurs, it's not something that we would have known if we didn't have fossils, and we would never be able to imagine this kind of variation. Even relatively, recent extinction can dramatically change how we think about diversity. So if we look at sloths, like in that bottom corner, just 5,000 years ago, 5 to 10,000 years ago, there was a diversity of giant ground sloths in the Americas. But of course, today there's just a few small tree living forms. And so if we didn't have that fossil record, we would have a very different understanding of how sloths evolved than what actually happened. Now, the other thing that we want to keep in mind when we think about the evolution of morphology or the evolution of form is the developmental systems that actually produce that variation or produce those forms. And it was stated, I think, very nicely by Conrad Waddington that the evolution of organisms must really be regarded as the evolution of developmental systems. So if we take this classic example in the middle of this slide here, of an axolotl and its sister species, the tiger salamander, these two forms look extremely different in their adult, sexually mature form. And so you might think, if you didn't take into account development, that there must have been a huge amount of evolution between these forms. But actually, we know, because we have information on how these species develop, that it's only a minor change in the timing of development that produces this incredible variation in form. So we want to try and incorporate that into our reconstructions of how morphology evolves as much as possible. Now we can try to do this by gathering direct data from developmental sequences. Like, so here at the bottom, these are some CT scans of a developing marsupial skull. And we can measure those skulls. We can try to reconstruct the exact, what we would call an ontogenetic trajectory of how the shape changes through ontogeny or through developmental time. But the problem is that a lot of those data aren't available for fossils or most species. So we have to try and think of how we can incorporate that. But it is especially important to consider because of this very important aspect, which is that these developmental pathways really limit the kind of variation that can evolve. And the way we can think about this is in gray dots in this figure on the left, the gray dots are if we simulate the evolution of form. So in this case, I started with a skull shape and just simulated how it would evolve if there were no limits or no constraints or no directions dictated. And it'll go and fill, kind of a spherical area of what we would call a theoretical morphospace that represents all the possible forms that could evolve. Now, if we add in developmental information, which the impact of that is to make certain traits evolve in a more coordinated way, what actually happens is that we see that many of those gray dots are never actually realized. But in fact, some of the forms that evolve are more diverse in certain aspects. So what development does is it forces evolution to go down a few relatively limited directions. And so it goes further down those directions, but other directions are never explored because of developmental processes and developmental constraints. Now we've called this model the fly in the tube model. And the tube is represents the area of possible forms that can evolve given a certain developmental pathway or developmental process. But within that tube, evolution can do lots of crazy, fast tinkering with forms, and that's the fly moving around this limited tube. And so some of the questions I'm really interested in is trying to understand how do developmental pathways really influence the tempo and mode of phenotypic evolution or the evolution of form. And then on a more broader scale, are some kinds of developmental pathways, are some kinds of ecologies or life histories, are they easier to evolve? Are they better at evolving than others? So for example, if you are an organism that goes through metamorphosis versus an organism that doesn't go through metamorphosis, does one of them evolve more diversity? Does one of them evolve faster in the face of environmental change than the other? And these are questions that are really important to try and understand as we move through this, kind of environmental and biodiversity crisis, because it will give us predictability for understanding how different species will respond to the changes in their environment. Now, this is all part of a larger project in my lab of trying to understand really what intrinsic and extrinsic factors influence the tempo and mode of evolution of organismal form. So intrinsic factors are things like development, maybe the genetic processes that give rise to morphology or anatomy. We can even think of ecology as an intrinsic process or factor. Extrinsic factors that would include things like the environment, maybe large-scale mass extinctions and things like that are outside of the organism. And so to do this, what we really need to do is gather a lot of data. We need to gather data that really captures the diversity of organisms and then try to reconstruct how those different shapes evolved, whether some of them evolved faster or slower, whether those shapes reflect ecology or life history, do they reflect habitat or diet, and how do they differ across some of the different groups that we look at. And so over the last 5 years, my lab and I have been traveling around the world building an immense data set to really span the diversity of land vertebrates or tetrapods. And so what we've been doing is using CT scanners and laser scanners to travel around to museums across the world and build a 3D data set for skull shape. And we've been focusing on the skull in particular because the skull has quite a lot of different functions. It's important for food gathering, food processing, sensory protection, for example, holding the eyes and the nose, the ears, and the brain, of course. As jaw musculature, lots of groups use it for things like fighting or for sexual display. So there's lots of different influences on skull morphology, which means it can tell us a lot about the various factors that really drive the evolution of form. And so far we have, about 1,900 species sampled. About a quarter of these are fossils. So we have a really nice representation of the fossil record. And because the methods that we use require perfect specimens, that's really quite, I think, an extraordinary data set. And over here in this big circle figure, you can see the phylogenetic tree or the evolutionary tree of the relationships among these other groups, showing that we have lots of amphibians, lots of snakes and lizards, lots of birds, dinosaurs, crocodiles, mammals, turtles, really the full range of vertebrate diversity, of land vertebrate diversity. And just recently, we've started a new project doing something similar for fishes. Now, what we do once we have these beautiful scan data is that we clean up our CT scans or surface scans, and then we use a combination of methods to really try and very densely or with a high degree of precision represent the shape of that skull in a way that we can then analyze. So to do that, we use discrete and curved landmarks. These are these points that you can see on the skull that end up looking like this. And so the red points are what we call landmarks that we can pick out on every skull. The curves then join those up to kind of delimit different bones or different regions of the skull. And then we use this in combination with a very generic template. And the template then gets mapped onto each of our skulls and gives us a very similar distribution that we can then compare across our entire data set. And so just to show those last few steps in detail, what we do is we take the curves and landmarks on every one of our skulls, and then we take a common template that then maps these blue points, these surface landmarks, onto each of our skulls. And so we can end up with a really, really nice representation of the skull shape across all of our diverse species. And so using this approach, we've applied this to all the scans in our data set. And you can see what we end up with is a really nice and really common way of, a really shared description of skull shape across a really wide variety of different organisms. And this is something that's extremely tricky, and it took us quite a while to figure out how to do it, but what we've ended up with is something that I think is very powerful. And what's nice about this also is because we use those curves to separate out the different parts of the skull, we can look not just at the entire skull shape, but different parts of the skull to see, is the brain case evolving the same way as, say, the mouth region? Or does the eye region evolve differently than, say, the neck attachments? And so we can start to look at how different parts of the skull might be responding differently to various factors of interest. And once we have that data set, we can start to look at the really interesting questions that we want to know. Specifically, what are these factors that are influencing how organismal form evolves and how diversity of organismal form evolves? And so this is an example of one of the papers, one of the first papers from this project that we published, looking at the evolution of bird skulls with a data set of over 350 species. And this, in the middle, this phylogenetic tree here, this is the tree of the relationships. And what we were able to do then is use the differences in their skulls and what we know about how they're related to then try to reconstruct which parts of the tree evolved really fast, which are the ones that have these yellow and orange and red branches, and which groups kind of evolve very, very slowly, which are the bluer branches. And what we do when we can do that is start to try and understand the factors that are really shaping why certain species and certain parts of the skull evolve fast or slow. So some of the interesting results that we were able to come up with our bird study in particular, to start with, was that diet, so what a bird eats, actually doesn't have a very clear relationship to what its skull looks like. And this is something that's really confused people for a few years now, because of course, when we think about Darwin's finches, it's a classic example of diet shaping the evolution of the skull. But when you look across all birds, that really doesn't hold, interestingly. So there isn't really a close relationship between diet and skull shape, but there is a close relationship between how fast skulls evolve and diet. So interestingly, the seed-eating birds and the nectar-eating birds are the fastest evolving. And that's interesting because, of course, that includes Darwin's finches or Hawaiian honeycreepers. Some of the kind of the famous adaptive radiations actually fall into that territory. So it actually seems that just by luck, really, the people who are developing these ideas about evolution happened upon the ideal birds to really figure out this relationship. And if they'd picked something like, some of the carnivorous birds, they probably wouldn't have had such a clear idea of how the natural world or the environment really shapes evolution. One of the other interesting things that we were able to look at by separating out the different parts of the skull is that different regions of the skull evolve at very, very different paces. And the fastest evolving regions of the skull are really the front, the rostrum, what we would say, the palate, and parts of the cranial vault. And what's interesting about that is those are all formed by the same kinds of developmental tissues, what we call the anterior cranial neural crest tissues. Whereas the parts of the skull that are developed by the mesodermal tissues, which is kind of a more ancestral developmental tissue, actually evolves quite slowly. And so we can start to piece together that maybe developmental tissue origin has quite an important role in the evolvability of different parts of the skull. Now, the other cool thing that we can do with this is also, because of our data set having lots of fossils, put birds into the larger context of their dinosaur relatives. And so to do this, we had a nice data set of not just of birds, but also of the non-avian dinosaurs, which of course birds evolved from. And what we were able to do then is compare how birds and other dinosaurs evolved. And so on these trees down here, you can see most of these trees are birds, but down here you can see the ones that don't go to the edges. Those are the dinosaurs. And very interestingly and unexpectedly, we found that dinosaurs on average evolve as much as 1.5 to 3 times faster than birds have since the other dinosaurs went extinct. So actually, once birds evolved, evolution really slows down compared to other dinosaurs. And what we think is happening there is that dinosaurs do a lot more different things with their skulls. They have these big fighting structures. They have these big display structures. They have much more complex feeding structures. They have a bigger diversity of feeding types. They have diverse tooth morphologies or tooth types. Birds, in contrast, actually really lock into one pattern. They have these big brain cases and their beaks, they lose their teeth. And so they don't really vary very much in terms of their jaw muscles. And they don't really, because they have to keep their skulls quite light in order to fly, they don't really add very much bony ornamentation or other structures onto their skulls. So actually, once other dinosaurs go extinct, birds go through essentially a variation bottleneck, and they never really recapture that kind of fast evolution, that really big kind of experimentation of different morphologies that other dinosaurs had. Now, this is interesting because we usually think of birds as a big success story, right? And the other dinosaurs went extinct because they weren't as evolvable or weren't as successful. But actually, other dinosaurs were definitely much faster evolving and evolved far more diversity than what we see in the living dinosaurs, the birds. And we can also look at other groups, and as we have. And so if we look at the squamates, the snakes and the lizards, using the same approach, we see many interesting things. First of all, we can look at all of them in a single kind of morphospace. So we still have some principal component analyses in the last figure in the last talk as well. So this is comparing the shapes of the different skulls. And oh, sorry, let me go back. Over here we have the snakes, and over here we have lizards. Here we have things like mosasaurs, the extinct forms that are related to them. And we can see there's a huge area of overlap in brown in the middle. And what that area really represents are all of the forms that have become burrowing forms, or the fossorial forms. And both snakes and lizards converge to a very shared skull type in doing that. And so we can compare these very, very different structures and show that there are really quite a lot of similarities in them. We can reconstruct their evolution as we did with the birds and see which of the groups really show fast evolution. And we see that whenever they move into an underground lifestyle, a fossorial or burrowing lifestyle, we see a burst of fast evolution. But interestingly, we see that different parts of the skull evolve very differently in snakes and lizards. So whereas in snakes, they're really the parts of the skull that are associated with feeding behavior are the ones, and the ones that are fastest evolving with dietary changes are the ones that are associated with opening the mouth. So kind of the back part of the skull where you open the mouth really wide to eat some of those big prey items are the ones that show the strongest signal of diet in that group. In contrast, if you look at lizards, the parts of the skull that really show that pattern are the ones that are the front of the skull, the biting parts of the skull, not the parts associated with opening the mouth. And so we see that despite being closely related and showing lots of similarities in skull form, the different parts of the skull evolve quite differently in these two groups. And then when we can look at amphibians as well, using, again, a very similar kind of skull data set, so we can compare the same sorts of patterns. And what we see, and this is an example of our data set, again, showing this really nice density of data across a diversity of forms. And what's really interesting is that what we expected to see in frogs is that development would have a very big impact because frogs go through this incredible metamorphosis, of course, going from tadpoles to their adult form. But interestingly, differences in how they develop, specifically whether or not they feed during that larval form, doesn't seem to have as strong of an influence on how frogs' skulls evolve as much as where they live. So the strongest impact or the strongest factor in what a frog looks like is what kind of environment the adult form lives in, not what the larval form is doing. And in fact, we found that the forms that live in the in aquatic habitats and in fossorial or underground habitats really evolve much, much, much faster than any other kinds of frogs. So really moving into the water or moving underground really means a fast evolving frog, whereas the other ones are quite slow evolving. Development, again, has some influence, though, in that the parts of the skull that evolve or that develop later in time. So some parts of the skull evolve or develop quite early during ontogeny or during development, and other ones kind of become bony much, much later. The ones that develop much later actually tend to be much more variable. So that suggests there are some kind of functional constraints on the evolution of the frog skull associated probably with their habitats. And then we can also look at our salamander group, and salamanders, I think, are maybe the most interesting of our data sets in many ways. An amazingly diverse group, both in terms of developmental strategies and in habitats. And interestingly, unlike what we saw, so gathering again the same sorts of data for the salamander skull, unlike what we saw in frogs, we find that development has the dominant influence on skull evolution in salamanders. So in salamanders, if you are a metamorphic salamander versus a direct developing salamander, which means that they're hatched from their eggs essentially in their adult form, or if you're a pedomorphic salamander like the axolotl that stays in its larval form throughout its life, these are the factors that really differentiate what their skulls will look like, and also how fast their skulls are evolving. So when we reconstruct the pattern of the rate of evolution across the skull, we see that those metamorphic forms, the paedomorphic forms evolve much, much faster and much more diversity of forms compared to the direct developing forms. So in this case, it does seem that metamorphosis really impacts how quickly you evolve compared to if you lose that. And they are ancestrally metamorphic. So losing their metamorphic stage actually really limits their evolution, which is quite interesting. Now, the last thing I want to mention is what you can do with these data, which we weren't able to do before, is start to look at what parts of the skull are evolving in quite a lot of detail. So in this skull figure up at the top, you can see there's some red dots and some blue dots and some green dots. Again, the reds represent really areas of fast evolution. The blues are areas of slow evolution, and the colors in between are in between. And so we can do this across all the species in our data set. So we've done this for about 1,500 species so far. And so there's some frogs, there's salamanders at the top, there's caecilians, which are these limbless amphibians, there's snakes and lizards and dinosaurs and birds and much, much more. And what we can see is that when we look at where the fastest evolution is happening in most of these groups, for most of them, the fastest evolution happens to be around the jaw joint, where the jaw meets the skull and the muscles that are associated with that, which are really important for feeding behavior. Now, what's interesting is that birds, and actually mammals also, although I don't have them on this figure, show a very different pattern. They show the fastest evolution happening in the front of their faces, at the tip of their snout or in their rostrum. So you see this kind of big separation across animals, across vertebrates, where you have some that show fast evolution of their jaw joint, and others show fast evolution of the front of their face. Now, the only ones that kind of go in between really are the theropod dinosaurs as they transition towards birds. So that's an interesting evolutionary story itself. But this raises a lot of interesting developmental questions that now we're trying to investigate. So hopefully I've convinced you that this approach, this kind of high density morphometric approach, gives us a really powerful tool for trying to reconstruct the evolution of morphology across really, really different kinds of organisms, and looking at both how intrinsic and extrinsic influences influence the evolution of morphological diversity. And what we can see is that both development and ecology are really important influences, but they have different importances in different clades. And so, for example, in frogs, it's more about habitat, and salamanders, it's more about metamorphosis. And we see that sort of difference across different groups. And that will give us a lot of information for trying to understand, again, how climate change and environmental change will really influence these different kinds of organisms going forward. Now, what we're trying to do now is actually build our climate directly into our data by looking at how climatic shifts have influenced morphological evolution. And so we can already see in some of these groups that the rate of change in temperature seems to be a primary driver of skull evolution. So that's obviously very interesting for the modern world. We're also expanding to new systems. As I mentioned, we're working on fishes. We're also working on new work looking at arthropods and trying to understand why there are so many insects. And we're also trying to develop new methods that will make this process go a little bit faster. And then finally, we're trying to build in direct data on development, looking at comparative ontogeny, comparative genomics, comparative transcriptomics, and also using these huge genomic data sets that are rapidly being generated across different groups to directly compare rates of evolution in molecular data and in morphological data, which I think will be really interesting going forward. So to close, I just want to thank my absolutely fantastic lab that make all of this possible, to the various funding agencies, and of course, all the people around the world that give us access to these collections. They're really critical for us being able to do this kind of work. And all of the scans, as we generate them, we put online, so they're freely available. So any of you could go and download any of these images and do whatever you want with them. Okay, thank you very much, and I'd be happy to take some questions.